During the chat, if you'd like, and if you want to ask questions in traditional format, you can unmute at the end of the presentation to ask questions as well. So Wilbur, you want to go ahead and introduce our first speaker for today?
Sure. Thanks, Deb. Thanks, everyone, for coming. It's my pleasure to introduce Dr. David Myers, who is now assistant professor in our Department of Biomedical Engineering here at Emory. And as you guys may know, David actually helped me start our lab when he came as a newly minted PhD, and he did his postdoc in my lab starting in 2011. And just starting in October of last year, he's now his own independent PI in the BME department, so we're very happy about that. And one of his projects remains based in hematology, so David still has an appointment with us in AFLAC, is really about platelet contraction and platelet biophysics, a project that I'm continuing to work with him, and very honored and glad that we can still collaborate on this. And I will now shut up and let David take over.
Thanks, Wilbur. I am thrilled to be here. Like we said, I'm a newly minted professor in biomedical engineering, and I'm thrilled to also be in AFLAC still. So I actually came to Wilbur's lab by a pretty non-traditional route. So I've always loved small things in mechanics, and some of you have seen this picture, but I actually remember the exact moment I knew I wanted to go to grad school. I was in an electrical engineering class in undergrad, and I saw this picture. Now, many of you may be looking at the bug, which itself is impressive. It's less than a millimeter in size, but what drew my attention was this really tiny gear train at the bottom. And what's amazing about this tiny gear train is you can use it to do things. So you can make a bug carousel, and you can also make the gears move. And wow, my videos are not working at all. And they were just a second ago. Just a quick question, Molly, do you have any insights, or did I literally just practiced this on my computer and it was great? No?
No, I mean, let me see if I can
see
this really quick. Hold on. See if that, you said you did try it already and it worked.
Yeah, I rehearsed this 3 times this morning just to make sure I'd get this right, and of course.
I wonder if it's because.
Hey David, if your RAM is running low, sometimes it'll do that. I don't know if you have other programs open, but you may want to close them.
Oh, okay.
The other thing, David, okay, looks like you might, yeah, why don't you see if that works there? What I've also tried, David, is getting out of presentation mode, and sometimes that actually works, but sometimes it doesn't.
What a bummer. I was so proud of this presentation because I had like, you know, a gazillion videos in here, and now you all get to see me hand wave my way through it. Okay, I do apologize for this. This is not the quality I like to present, but moving forward, I'm just going to continue with the presentation. My apologies. Anyway, you can actually make these things move. This is a bug carousel, and this gear train actually moves, and you can still see it at the Sandia National Labs website.
David,
Now,
if you think it might be helpful to shut down PowerPoint and restart it, take a minute to do that. These type of technical glitches happen in these virtual seminars all the time.
Okay, I do apologize.
They can take a minute to answer an email.
Okay, thank you. I'm going to try one more time. I do
Wait, Deb, are you saying you don't think every single person's paying 100% to David and not doing any email at all? I don't believe that.
I'm sure they are, especially after seeing your office background. I'm sure everyone is 100% riveted.
All right, let's see.
It's okay, David.
Well, it's, you know, Wilbur, you've always given me the advice to show as many videos as possible, and I was so proud of this presentation because I managed to get one every like 3 or 4 slides.
Yeah.
And
right.
of course, I don't know if Alisa is online. I'm sure she's laughing at me a little bit.
Right. Well,
Animations. There. Okay. Can you guys see the animations?
there you go. All right.
Okay. All right. Thank you. Thank you for bearing with me. I will try to make up the lost time. So you can make a bug carousel, and you can make a moving gear train, and you can still see this. Now, if we think about my interest in small moving mechanical things, that actually translates really well to biology, eventually with a lot of practice. And what I'd really like to focus on is what Wilbur and I have spent a lot of time studying together, which is hemostasis, and specifically the end stage of hemostasis, which involves this contraction phase. So we know that at the very end of hemostasis, that you have, we all know about how hemostasis happens. There's platelets and fibrin in this clot, and eventually it contracts. Now, if I take platelets and fibrinogen, the precursor to fibrin, and thrombin, the molecule that both activates platelets and converts fibrinogen into fibrin, and just throw them in a tube, you can actually see that the clot contraction process is inherently mechanical. It goes through an order of magnitude reduction in size, and it actually is increasing in stiffness by an order of magnitude. And this dramatic process on the left of my screen is actually mediated by platelets in red pulling on a fibrin mesh in green. Now, you may be asking yourself, yeah, so what? Does this even matter? And the literature suggests that it does matter. It seems that the mechanical properties of blood clots do have an effect on hemostasis and thrombosis. For example, in young patients that had a post-myocardial infarction, we've taken their clots and we've seen that these clots are 50% stiffer and more resistant to dissolution as compared to healthy controls. In patients with bleeding disorders, their clots can be 7 times softer, and they're also more prone to dissolution than healthy controls. But the question is why? What in the world is going on? And you can see in this picture above from John Weisel's group, it's a beautiful false colored SEM showing platelets in purple, fibrin in blue, and occasionally entrapped red blood cells, that there's a lot going on in this active biomaterial. Now, from a mechanics perspective, what we need to do is understand the individual components and how they interact to ultimately come back to answer this question of what dictates blood clot mechanical properties. Now, from the material that you saw in blue, fibrin, it is a complex material, but it's very well characterized. We know that it's a nonlinear material. We know that its structure varies with flow and different agonists, but these things can be modeled. We can actually, as long as we have good, solid experimental data, we can create an experimental model based on fibrin that incorporates fibrin. Now, many of you know that I have been working for a number of years with Wilbur on understanding the platelets. We knew a lot less about platelets when I started my postdoc. And especially, we didn't know much about the forces that individual platelets could apply because we didn't have the right tools. So Wilbur and I worked together and we made this really amazing system that we're both quite proud of, where you actually have these simple micro-patterned dots cross-linked on a hydrogel. The hydrogel is movable. A platelet can land on one of these fibrinogen dots. They're covalently cross-linked to the surface, and as the platelet applies force, it moves the dots, and the microdot displacement is proportional to the force. And what you can see here is these are the dots, and this is a moderate and highly contractile platelet. And what's been wonderful about this system is it started to give us the data that we needed to start creating full-scale models of blood clot contraction. For example, we actually mapped out the contractile force response of platelets for healthy individuals. Now, this is a range of thrombin concentrations that may be seen within a clot. And this is the range of stiffnesses that might be encountered within a clot. And what you can see is that platelets have a pretty dynamic response. In low thrombin concentrations and low stiffnesses, platelets apply low forces. And there's this synergistic middle where platelets can apply a very high force. And that as you increase the thrombin concentration or as you increase the stiffness, platelets apply less force. Now, just as a quick aside, I'm sorry to do this, but I just want to, bring it up because we're talking about platelets, which are near and dear to my heart. We've actually been looking at platelet force as a biophysical biomarker. Many of you have seen some of my work with Wilbur on immune thrombocytopenia, And what we found is that single platelet forces do seem to correlate with bleeding, strongly correlate with bleeding in immune thrombocytopenia. So higher Buchanan bleeding scores, which indicate more severe bleeding, all have low platelet forces and low platelet counts. And this has been remarkably high sensitivity, and when we consider count, remarkably high specificity. And now what we're trying to understand is we see a correlation, and we're trying to understand the causes of low platelet force and see if that gives us any new insights into ITP. So that was my quick plug. And now what I'm going to focus on for the rest of the talk is a new story that I want to share with you. Now, I showed you this video at the very beginning where you can see this bulk clot contraction event, and you see this dramatic size change in the clot. And what we also know is that single platelets are mediating this event on the right, but we don't, what in the world is going on? That is a big jump between these macro scale clots and these micro scale platelet movements. So this is what the rest of the story is about. Wilbur and I and this really amazing group in mechanical engineering set out to really look into this process and understand what's going on. Now, the place we started was just collecting a lot of experimental data on, what are the final sizes of clots? What are the kinetics of clot contraction? And so what you can see is a typical experiment that I did with platelet-rich plasma with thrombin, threw it in an incubator, taped my iPhone to the side of the incubator, and watched what happened after a couple of hours. And you can see that I've gotten better at making these clots. And one of the things that really struck me was the clot really maintains the shape through the entire process. It's a cube at the beginning and it's a cube at the end. And I think I did what any of us would have done in this case. It sparked my curiosity, and I went to Michael's Arts and Crafts and I bought every single chocolate candy mold I could find, and I made all kinds of crazy clot shapes because I wanted to understand what was the fidelity? Were we always going to see these shapes? So here's a Lego person contracting for your viewing pleasure on the right. You can actually see that the Lego person starts out as this large clot, and the features are very well preserved throughout the process. And like I said, I bought every single candy mold I could find, so you can see a Lego person, you can see a seashell, you can see a sea star, and also because we're friends, I'm showing you the less optimal versions where I did a puzzle piece and also the letter A. And I was really fascinated by all of these because you can see that there is a high level of detail preserved as the clot shrinks down. And even holes can get smaller as we continue to contract. Now, as an engineer, putting on my engineering hat, I'd love to know if I can make an analytical model. Can I just come up with something that's good enough that would explain this process? So what you can see is this is our experimental data in gray. And the first pass at a model that we came up with, Alex and Yuyi, who are over in mechanical engineering, was we assumed that the platelets were evenly spaced within a clot. I realize that's an assumption, so I'm, just full disclosure. And we also assumed from our individual platelet studies that each platelet had this radius of influence, an area around itself that it could contract that was set by Filippodio length. And we assumed that these platelets all contracted and pulled this clot in. And what we found was that the analytical model that we came up with was horrible. It didn't match at all to the experimental data. And, of course, that's always a little crushing. It took a little work, and your numbers are so off compared to what experiments are showing you. But when you take a step back and you look at what did this model assume and what might it have been missing? This model assumed that every platelet worked on its own, independent of itself. So the first step is maybe asking the question, well, I don't know if that's true. They probably do work together. And so the challenge becomes to work together, you actually need to make a full-scale, pretty serious clot model. And that was our first step that we all did together. So what you can see here is this is our a single, we call it an in silico platelet or an iPlatelet, and we put it in this iFibrin mesh It has I-Philopodia. Of course, we can carry this all the way through. And you can see that when we look at this platelet from the initial position, it can send out these Philopodia, and it wraps itself in fibrin over time. And this seems to match what we experimentally observe with scanning electron microscopy, where the final platelet shape is round and kind of wrapped in fibrin. We also took a clot, the same approach that I mentioned earlier, where we took fibrinogen, platelets, and thrombin, threw them in a dish, and quickly fixed it and ran over to the confocal, and we got a digitized fibrin mesh that recapitulates what an experimental mesh would look like. And now we have the base components of a model that we can use to understand clot contraction. So going back to these videos, you can actually see that over time, this I platelet wraps itself in fibrin on the left. And you can see that this seems to recapitulate what an actual platelet is doing on the right, which is wrapping itself in, and it's using its Philopodia, pulling out and wrapping itself in a mesh. So now the question becomes, we have all of these components. We have I platelets, we have a mesh. Let's put them all together and see what happens. Now, before I do that, it's always good to go back to experiment and ask the question, well, what do we expect to see? So going back to our clot in a dish, where this is a 3D view of, this is the top view of a 3D clot. So I'm kind of looking down, or actually in this case, it's confocal, so I'm looking up through a clot. And what you see is platelets in red and fibrin in blue, and I'm just gonna let this play. And you see that two things happen. First, an emergent behavior that happens is that the platelets themselves cluster. They draw together. The other thing that we see happens is that between clusters of platelets, the fibrin is actually thickening. So what we were hoping to see in our model is that maybe this behavior would emerge. And to our delight, it did. We actually do see this behavior. So let me walk you through this graph. This is our initial clot. And this was the final contraction of this clot here. And if we zoom in on this clot, we can actually see that there's clustering and alignment of fibrin. Now, this model was a very positive step in the right direction. You can see that it is much closer to meeting our final clot contraction volume. It contracts much more than our analytical estimate. But there's still a lot of room for improvement. And I know that between 30 and 10%, it may not seem like a lot, but the details matter in this case. Getting that last 20%, that's, it's 3 times different at this point. And we were really kind of racking our brains saying, well, what in the world could be different? Why is this happening? And what we started to look at was this timing difference. When we initially started this project, we actually had all of the platelets contract within 15 to 20 minutes. However, we can see from experimental data that platelets contract over a much longer time frame. And this kind of gave us a hint that maybe the timing really matters. So what I'm going to show you here is we know from previous studies that platelets are heterogeneous. We know that they're heterogeneous in size. Our studies have showed contractile force. The expression levels of p-selectin, phosphatidylserine, and alpha-2b beta-3 are different. But what about the timing? And when we look at this kind of same 3D clot in a dish, and we look at phosphatidylserine exposure, which is an end marker of platelet activation, we actually see that there's a lot of timing heterogeneity, and that this is very consistent among different donors and for a lot of different clots, and that we see that this number of end-stage platelets, as measured by PS, steadily increases with time. Now this video, that showed us what's happening in bulk, but what we don't know is what's happening at the individual platelet level. Now this video, I will admit, is a little hard to find. So what I'd like to do is draw your attention to the Philopodia. These are 3 platelets that were in the same frame, imaged at the same time, so that these are synchronized frames. And what you can see is that the filopodia of these individual platelets are extending and retracting at different time points. A single platelet will extend and retract filopodia at different times unto itself and also compared to one another. So this is kind of letting us know that maybe the timing matters. And how can we possibly incorporate this into a model? And the way we decided to incorporate it was the following. The first was, when do the platelets begin their contraction process? So you can see here that this platelet at frame 0 has already started contraction, and these two haven't. And as we go along with time, these platelets, each of them, will start contracting at different times. So this one's already started, this one starts here, and this platelet starts at this time point. So that's one way to model heterogeneity in timing. The other way to model heterogeneity in timing is, when do the filopodia themselves extend? And so what we have is, we have waves of filopodia. So a platelet can extend filopodia, extend more filopodia, and then finally finish contracting. Now, just to remind you, our experimental data, the final clot volume is about 0.1 as compared to the original, so 10%. When we have all of the platelets synchronized together, the final clot volume is about 30%. What's interesting is, when we combine both of these features together, when do the platelets start contracting and this filopodia heterogeneity, that's when our model starts to match our experimental data. Now, you may be wondering, how does this work and what is the process? And I think that if we just consider a really simple system of 2 platelets and fibrin, it'll make a lot of sense. 2 platelets, A and B, if they contract synchronously, they immediately send out filopodia, grab this fibrin, and just pull it together. However, if we can insert a pause, if one platelet can reach and the other waits just a little bit, and there's heterogeneity, the platelets can actually work together and bring fibrin much closer together than if they had just pulled and reached at the same time. Now, the other thing that this asynchronous behavior does is it allows us to begin to match the kinetics of clot contraction. The number of initially contracting platelets, so here this is 10% of the platelets contract and then the rest contract over time. This is 50% of the platelets contract initially and then the rest contract with time, and this is 90%. And when we get to between 50 and 90%, the kinetics of the clot contraction match pretty closely with experimental value. So you can see that there's this initial phase of very dramatic clot contraction and then a slow clot contraction with time that continues over 90 minutes. And what's also fascinating is that the ratio of platelets that initially contract, how many of them just turn on and contract at the very beginning, it doesn't strongly affect the final clot contraction volume, as shown here, but it does really affect the kinetics. Now, one thing to just keep in mind is that all of the data that I've shown you is for a single platelet concentration and for a single situation. And modeling people and experimentalists tend to argue that you can make anything fit one defined set of computational numbers. But the question is, can this be expanded and applied to other areas? And what we found was that if we take this fibrin mesh and we just change the platelet concentration, the first thing that we found is that there is actually a maximum clot contraction point. So when you get to a certain density of platelets, you strongly contract the clot to a really small value. And as you add more and more platelets, they actually get in the way. Their bodies physically take up space, and you can't get smaller than this contraction volume. And what's remarkable is over 3 orders of magnitude of platelet concentrations. You can see our experimental data in black, and our model with asynchronous platelet contraction, where there's pauses and timing and waiting, we actually have pretty strong agreement with our experimental data. And then you even have this match in the maximum clot contraction and then subsequent increase in the clot size as you add more and more platelets. And just for comparison, this is what happens with synchronous platelet contraction. But again, this is a model, and what's fun about models is you can do stuff that you can't do experimentally very easily. So we can actually start to understand what's going on inside of a clot as it's contracting and how it applies force. And so in this particular case, we held the clot between two fixed surfaces and allowed it to contract. And what I'm going to draw your attention to over here is this is the first time that an experimental clot model with forces that were akin to what we see in platelet contraction cytometry matched experimental bulk clot contraction values. And that only occurred when the platelets behaved asynchronously. Because it's a model, we can also force all of the platelets to contract at the same time initially and see what the difference is. And I realize this may be a subtle point, but I think it's important. When you look at the increase in the amount of force that the platelets have applied here, it's a 70% increase in force, and the only thing the platelets are doing differently is waiting. Inserting pause points is what allows these platelets to actually increase the total amount of force that they apply. I'm running out of time because of my technical glitches, but I just want to say that this can actually be generalized and that we can actually look at, we've made analytical equations and expressions and have wonderful matching with physical systems. So I'd like to just close with a couple of high-level thoughts. The first is that mechanobiology is traditionally thought of in terms of space, 3D. But this work really shows that timing matters a lot, and we really need to think about it in 4D. And specific to platelets, platelets having timing differences actually leads to smaller final clot contraction volumes and increased force generation. We're excited because this model is that link between single platelet behavior and bulk clot mechanics, and it may help us to understand altered clot mechanics in various pathological disorders. But there's another interesting point. This is showing that platelet heterogeneity, which is often, I think, thought of as an artifact, at least in my mind, may actually serve a biological function. At least from a biophysical perspective, heterogeneity actually seems to enhance the function of the collective of platelets. So with that, I'd like to just close. Again, I worked with a really amazing group of people. This was a really fun project. Wilbur, it's always a pleasure, and I love working with you, and it was really fun to work on this project. And we're submitting this paper now, and we're going to see where it goes. Alex and Yugi and the complex fluids modeling and simulation group and mechanical engineering are amazing. They've asked me some of the most fascinating questions I've ever gotten. And if you ever have the opportunity to work across disciplines, I would highly encourage you. And then I'd also just like to give a shout out to Mo, who I'm really lucky to I get to co-advise with Wilbur, and also my new lab, and also our funding sources. So with that, I'm going to close and take any questions. Thanks.
Great. Thank you, David. So Deb, maybe we have time for 1, 2 questions?
Yeah, we have time for a question or two.
Sorry.
All right.
Hey, David.
I'll jump in. Go ahead, Curtis.
Oh, go ahead, Clint.
No. You first.
Yeah, I'll make this quick. So, David, fascinating work, man. You know, I'm a big fan of what you guys do with the functional and biomechanical studies. So I was wondering if you know if mitochondria or ROS play a role into the timing or force, because, you know, platelets don't have nuclei, and we know at least with aging and stem cells, if you have defective mitochondria or deregulated ROS, it can change everything, usually in a pathological way. So have you guys looked at if the mitochondrial load or amount of ROS you see in these cells have any impact on timing or contractile strength or anything of that nature?
You know, that is a great question. We have looked at mitochondria a little bit. We haven't looked at it with regards to timing. We have looked at it with regards to force. And so we had kind of 2 running hypotheses. The first was that the number of mitochondria might seem to influence the force because there's a lot of heterogeneity in the numbers of mitochondria per platelet. And we saw absolutely no correlation between force and the mitochondria number. And I'd have to double check my notes, but I'm even pretty sure that we tried mitochondrial inhibitors. I'm a novice in this area. I don't remember which ones. And we also did not see a force difference, which we found really surprising. But I'd love to keep chatting with you about this and see if there's something to work on.
Thank you.
Yeah, if I might, fantastic talk. Really enjoyed it. Just to pick up on that, do you have any sense of whether or not platelet age has anything to do with the activation time or whatever you want to call it, the heterogeneity in time?
That's one of those light bulb moments. The moment you said that, I thought, oh, right, I should have looked at that. I hadn't thought about that, to be honest, and that's a great question. And I'd love to see if I can explore that.
Well, we do, David, have some of that data, at least regarding force, right? Like the reticulated platelets, right? We didn't really find a specific correlation, at least with force. But it's a good point, both the Curtis's and Clint's questions. Timing may be different than force, right?
And I would also consider looking at the heterogeneity in your different disease contexts that you mentioned in the first part of your talk. I wonder if heterogeneity in those contexts actually contributes to those diseases. But it's a thought. We see it in hematopoiesis, but I'm going to shut up, I promise.
Yeah, but I do think, David's studies are really neat. And, all of us, at least in the hemostasis and thrombosis fields, know that platelets are very heterogeneous. We don't know why. And here, we're getting at some semblance of the question, well, maybe this is why, right? There's one example here where having things be very different, having different populations that turn on different times may actually matter and may actually contribute to function. Yeah. All right. Thanks, David.
Thanks, everyone. Sorry, I'm a little over.
Thanks, David.
So I will introduce our next speaker today. It's going to be Ray Hunter. Ray Hunter is a fourth-year graduate student in my lab. She came to us from Georgia State, where she not only earned her undergraduate degrees, but actually a couple of master's degrees, including in biology. And she's already made some important contributions in our lab, contributed to a paper that we published last year in Cancer Research, and was recently awarded an F31 training grant. And today she's going to talk to us about how myeloid cells contribute to the control of leukemia, or rather how leukemia cells evade immunity during leukemia development.
All right. Thank you so much, Chris. Can everyone see my slides? All right. Once again, thank you, Chris, for that introduction. I am excited to discuss with you today my research regarding myeloid cells' contribution to early B-cell acute lymphoblastic leukemia clearance. And I'd also like to thank my advisors, Dr. Christopher Ford, and also Dr. Curtis Henry, for their input and direction for my project. So first of all, why even investigate immune evasion in childhood leukemia? Leukemias are the most common cancer in children, and unfortunately, they still remain the second leading cause of disease-related deaths in children. And more specifically, acute lymphoblastic leukemia accounts for 75% of all pediatric cases in the United States. And although current therapies have developed that have improved therapeutic outcomes in patients and also shown the potency of the immune system, such as CAR T-cell therapy, there still remains a need to further understand immune evasion during leukemogenesis. So previously, as Chris described, we recently had a paper published in Cancer Research that reported knockdown in calcineurin in leukemia cells leads to leukemia clearance in wild-type mice. And as you can see here, for percent survival is represented by this red line. This is in your wild-type mice engrafted with calcineurin-deficient leukemia, exhibiting prolonged survival, and also represented on this left panel is reduced leukemia burden. However, in TCR alpha null mice, which are deficient in T cells, you do not see a similar trend in which calcineurin-deficient engrafted mice actually have a reverse in percent survival, reduction in percent survival, and also an increase in leukemia burden. And so based on this data showing that the importance of T cells in leukemia clearance, we wanted to identify which T cell subset was potentially responsible for this leukemia clearance that we see. So I set up an in vivo experiment in which I depleted CD4 positive T cells, or either CD8 positive T cells, or both in combination. And I saw that regardless of which T cell subset was isolated for depletion, our mice had a reversal in this trend of survival and actually increased in leukemia burden. And so once again, this exhibited the importance of T cells and leukemia clearance in our calcineurin deficient model. And so additionally, to begin to tease out these mechanisms of calcineurin immune evasion, the Porter Lab engaged in various lines of investigation. And one such investigation was to explore cytokines and chemokines secreted in our calcineurin deficient leukemia compared to our non-silence control. And so what we saw on the left panel, you have your interleukins. On the right panel, you have your chemokines. And what we saw here, we had several hits. We saw that IL-12P40 was secreted at higher levels in addition to IL-13 and CXCL5. When we conducted further validation via ELISA, only IL-12P40 was significant. And we saw that regardless of cellular concentrations plated here, your calcineurin deficient leukemia secreted IL-12 at much higher expression compared to your control and also your parental leukemia. And with this interesting piece of data, we decided to go in vivo and do recombinant IL-12 treatment, grafting our mice with actually an aggressive BAL, and to see what would be the result of that. And so mice were dosed from day 3 to day 17, and then discontinued dosing after that. And as you can see, interestingly, your wild type mice, which do have an adaptive immune system intact, with just recombinant IL-12 treatment, these mice experience prolonged survival even upon rechallenge. And what's even more interesting is that in your RAG1 null mice, which do not have mature B or T cells intact, their control counterparts succumb to this leukemia. However, we saw a prolonged survival in our RAG1 null mice that had this recombinant IL-12 treatment, as shown here. And so we thought this was very interesting. And based on this data, we concluded that there was likely some IL-12 responsive innate immune cell population that was responsible for this prolonged survival, significant prolonged survival we saw in our RAG1 null mice. And so just a brief overview of interleukin 12 or IL-12. It is a pro-inflammatory cytokine that's naturally produced by your innate immune cells, including your monocytes and macrophages, neutrophils, and dendritic cells as response to pathogens typically. And IL-12 induces proliferation and enhancement of cytotoxicity, and also the production of cytokines, particularly interferon gamma. And in this context, interferon gamma in your natural killer cells. And so this was a line of investigation that we wanted to further explore, considering the effect of IL-12 on this population. So one of the first questions I had was, are natural killer cells significant for this leukemia clearance that we see in our calcineurin deficient model? And so I subsequently set up an in vivo experiment engrafting mice with calcineurin deficient leukemia. And I did this in both a wild type mice model and RAG1 null model. And in panel B, you just see a representative figure of verifying depletion. I targeted NK1.1 for depletion for our natural killer cells. As you see here in panel C, these are the results of my in vivo experiments, in which both your wild type groups, isotype control or NK1.1 depleted group, pretty much had a leukemia clearance compared to both my RAG1 null groups, which progressed in their leukemia burden. And this is also represented here in their percent survival. And so to kind of corroborate with this data, I looked in vitro as well. I did a co-culture for our natural killer cells, and in conjunction with either control leukemia or calcineurin deficient leukemia. And as you can see here, regardless of ratios in which our natural killer cells were plated with either control or calcineurin deficient leukemia, they pretty much had they pretty much had similar killing in your natural killer cells of either leukemia. And so this led me to conclude that natural killer cells are not critical to the control of BAL leukemia clearance, as we previously hypothesized. And so subsequently, I wanted to look into if myeloid cells played an important role in early leukemia clearance based on my previous data regarding natural killer cells. And just a reminder, once again, we wanted to evaluate myeloid cells in early leukemia clearance, considering these cells actually do secrete IL-12, and IL-12 has been shown to act on these populations as well, in addition to their important role in priming the adaptive immune system. And so I conducted another in vivo experiment grafting mice with either control or calcineurin deficient leukemia cells, and looking at very early time points. Previously in lab, we've looked at later time points, day 5 or past day 5. We wanted to see even earlier what was going on, considering the population I was interested in looking at. And so we saw that even by day 3, you see that there's this striking difference in leukemia burden in control leukemia versus calcineurin deficient leukemia engrafted in these wild type mice. And additionally, I verified this in the bone marrow, these RGFP positive leukemia cells, and I saw that there was over a 2-fold percentage increase in GFP positive leukemia cells in the bone marrow of our control mice compared to our calcineurin deficient mice as early as day 3. And this is also quantified here on the right. So this is the first time our lab verified the presence of these leukemia cells in the bone marrow at this early time point. And you see a difference even in the absence of calcineurin, that like in the absence of calcineurin, showing you that innate immune cells are likely apparent here and playing an important role considering the striking difference you see at such an early time point. And so to kind of go into this further, I assessed different myeloid cell populations. And first I looked in vivo at our dendritic cell populations. And I saw that utilizing CD11C positive marker for total dendritic cells, there wasn't a significant difference in your total dendritic cells from day 1, 2, and 3 in either our control or calcineurin deficient engrafted mice models. However, looking at activation using CD40 positive marker in dendritic cell populations, you see at day 1 that your mice engrafted with calcineurin deficient leukemia had an increase in DC activation. And then this trend is reversed at day 2, which is likely implicating some kinetics of CD40 and earlier activation of your dendritic cells and calcineurin deficient engrafted mouse model compared to the control. And so DCs have a more immunostimulatory profile in these mice that are engrafted with calcineurin deficient leukemia at an earlier time point. I also assessed other myeloid populations, including neutrophils and macrophages, and did not see any differences in activation markers. And this data is not shown. So subsequently, I wanted to also look in vitro to see if this would corroborate with my previous in vivo data, and based on the immunostimulatory profile that I observed in our dendritic cells. And so I did a co-culture isolating dendritic cells from mouse spleen, and I either cultured them with control leukemia or calcineurin deficient leukemia. And what I observed was that there was an increase in my total DC numbers, representative here, in my calcineurin deficient leukemia compared to the control. And when looking at activation markers, CD86 and also CD40, you see a slight trend, an increase in these markers, but nothing significant between the control or calcineurin deficient leukemia. Additionally, then I wanted to also observe if what would occur in vitro with our macrophage activation in our control leukemia versus our calcineurin deficient leukemia. And I utilized a ROD264.7 cell line, which is a murine macrophage cell line, in order to conduct these experiments. And what I saw was that they're using CD80 as a marker of activation, also CD86. There was no significant changes in activation in our ROD264.7 cells cultured with any leukemia type, whether it's parental or calcineurin deficient leukemia. And this coincided with what I saw in vivo as well. However, interestingly, once stimulated with LPS, you get increased activation in these ROD264.7 cells. However, there is a reduced activation in the same cells when co-cultured with leukemia cells. And so this is a very interesting find to see that when your ROD264 cells are co-cultured with leukemia, they're able to elicit the same stimulation, same activation as seen with just LPS alone. And so this led me to my next investigation, which was to identify the role of these myeloid cells in leukemia clearance in an in vivo model. And so I targeted colony stimulating factor 1, which is a receptor that mediates the production, differentiation, and function of myeloid cells. And what I saw, interestingly, these mice were engrafted with calcineurin deficient leukemia. And if you remember from previously, mice, wild type mice that are engrafted with this type of leukemia are able to exhibit a clearance of this leukemia and prolonged survival. And in depleting these myeloid cells, I saw a reversal of this phenotype. And these mice actually experienced an increase in leukemia burden, contrary to what we had previously seen with using this depletion antibody. And so I depleted these myeloid cells earlier in engraftment, and throughout, these mice are unable to control leukemia as a result. And so based on this, I wanted to see what impact do these potential professional, antigen presenting cells have on actual T cells as a result of this depletion. And so looking in T cell numbers from mice that were depleted in this study of CSF1R, I saw that your total T cell populations from your isotype group to your wild type CSF1R depleted group have a striking difference in total T cell numbers. There's a significant decrease in your CSF1R depleted group. And I did not see as significant of a trend in the spleen. And further, drilling down further into these T cell subsets, what you see that is in the bone marrow, both CD4 positive T cells and also CD8 positive T cells are at a decrease in the number of them in the bone marrow. And looking at the spleen, I do not see as significant of a trend, but you do see in CD8 positive T cells this significant increase as well in your CSF1R depleted group of mice. And so around this time, we were grounded from research, unfortunately. And so based on that and the data that had already acquired, we wanted to look into potential mechanistic factors that could be secreted that are altering our immune environment. And so based on previous data that we had in the lab, I evaluated that for our immune suppressive markers for the cytokine array. And what I saw was that osteopontin was secreted at higher levels in our control supernatant for our leukemia. And osteopontin is a glycoprotein that's been implicated as a mediator of immune suppression. And in further mining the data via OncoMine, I saw that SPP1, which is the gene that's encoded by, is expressed significantly higher in BLL compared to your PBMCs. And in looking through literature, I found that OPN has been associated with macrophage polarization, leading to more immune suppressive phenotype in your macrophages. Also implicated in leukemia cell dormancy and identified as a prognostic factor in AML and also potential prognostic factor in childhood BL subgroups with its different isoforms of OPN. And so this now leads me to my working hypothesis that osteopontin is a critical mediator of immune suppression in BL, and that this OPN, CSF1, CSF1R axis plays a critical role in the immunosuppressive microenvironment in BL. And being able to block this axis will enhance the efficacy of IL-12 treatment in conjunction with potential checkpoint inhibitor treatment in BLL. And so this is my working model that I've created, in which OPN is a critical mediator of immune suppression. And so you have potential secretion of OPN from these BL cells, and then this secretion of CSF1 is resulting in this autocrine signaling that's leading to a M1 to M2 polarization in your macrophages, leading to immunosuppressive cytokines and inhibition of your CD4 positive T cells and CD8 positive T cells. And you can reverse this phenotype targeting CSF1R and potentially some other targets in this working model. And so just conclusions from what I've previously discussed, NK cells do not play a significant role in our calcineurin deficient leukemia clearance. Additionally, our myeloid cells appear to be critical for early anti-leukemia immunity, and depleting these cells results in a decrease in your T cell numbers in both the bone marrow and spleen by day 12 in an in vivo model. And also, leukemia cells have reduced activation of our stimulated macrophages in vitro. So I have a long laundry list. This is just some of it to further explore this. But now I'm able to go back to lab, which I'm very excited about. And one of my first experiments to do is to evaluate secretion of OPN in our murine and human BL cell lines. I also want to explore this neutralizing OPN in co-culture and examining if there's this macrophage shift and change in cytokine chemokine secretion. I also want to, of course, do some in vivo studies neutralizing OPN and also doing a knockdown OPN in our BL to see the influence on leukemia burden and survival, and then explore potential for combination therapies to increase immunogenicity of our leukemia environment and hopefully have some clinical translation research in doing these explorations. And so with that, I would like to thank the Porter Lab. Dr. Porter has been great during this time. He made sure he set up all our meeting schedules still and checks in with us. And we are still keeping the science going even when we've been grounded. And just all my lab colleagues, they're amazing. I love being in this lab. Everyone's very supportive and there for me and to help me grow as a scientist in training. And I would also like to thank Dr. Henry for just his continued input and direction and encouragement, and also checking in, making sure that as one of his grad students that I'm okay. And I would like to thank my funding sources. I am a recent recipient of F31. I found out right before we were actually grounded for going to lab. So that was pretty exciting. I'd like to thank our other funding sources in the lab, and then Emory Winship, AFLAC, of course, Emory IMSD program, and Laney Cancer Biology Graduate Program. And at this time, I'll take any questions.
Thanks, Ray. So we do have time for questions, and I do want to reiterate that Curtis Henry is a co-advisor for Ray, and therefore I will defer all hard immunology questions to him.
Thanks, Chris.
You're welcome.
Ray, can I just ask what the sort of timeline is for your project in terms of writing it up? This new work is it's a fantastic use of your time away, but it's really quite a turn, and I can see where it could blossom into a complete another project, right?
Yeah.
It's your intention, and so I'm just wanted to check in and see what your thoughts are about that.
I anticipate, I've spoken with my advisors about it, and we still need to talk and kind of go through some more experiments, but I anticipate a year likely for getting it to the level where we feel comfortable to submit for a publication. And I do have other previous data, some other storylines that are interconnected that will kind of feed into this storyline as well. So I'm not going to go too far down the rabbit hole with this, but yeah, definitely it'll take some time, but I don't know, being out of lab too, it just gives you even more passion. Like, I can do this, you know, and also what's feasible. And I know both Chris and Curtis are great at making sure I do whatever's in the realm of reality more so than my loftiness sometimes going off and thinking of so many other directions. But yeah, probably around that time.
It's nice to see how the project is developing.
Thank you.
Okay, well, thanks everybody for joining, and hope to see everyone soon.
Thank you. Have a good day.
Thanks, Ray.